So I will talk today about mechanosensing and mechanotransduction at the endoplasmic reticulum. This is a view from a mountain over Innsbruck and probably our institute. I'm very bad in geography, would be somewhere here hidden. That's the river that crosses the city. It's really beautiful. You should come and visit, especially in summer when there's no snow. City is much more lovely than in winter. So my lab has been studying, it started as a postdoc, and then when I start my own lab, we are interested in signaling at the secretory pathway. And there's a lot of signaling that has been identified by my lab and several other labs. And this is signaling by mitogens, growth factors, amino acid starvation. And so these are biochemical stimuli that come from outside the cell. There's also like needs from inside the cell, like protein folding or misfolding, mitosis, which would be an intracellular need that would modify the ER, the exit from the ER. I'm not just speaking about exit from the ER, but in general about the ER architecture and function. And what has been missing is, mechanical stress has been overlooked in terms of, how it regulates the secretory pathway. So we decided a few years ago, and I will just show now, like, the take-home message from this published work, because the rest of the talk is totally unpublished data. So we showed that when we stretch cells, when we expose them to mechanical strain, we get a signaling by RAC1 that dimerizes with SAR1, the GTPase that initiates COP2 vesicle formation or COP2-dependent export from the ER. And this triggered the formation of new ER exit site and accelerated ER to Golgi trafficking. And why I'm showing this cover is because I was very proud. I said, this is like an accordion. You are stretching the ER, and then it makes COP2 vesicles. And while I was showing this to someone, actually, I noticed that this cover is wrong. Yeah. That what we showed is actually not stretching of the ER, we showed stretching of the entire cell, and then there's signaling to the ER exit sites that make COP2. These are vesicles. I know some people here oppose the idea that there are COP2 vesicles, but this is just a cartoon. So when I had this wrong cover in mind, I said, okay, but actually this would be a very cool question. Is the ER itself sensing mechanical stress? Like, is there a mechanosensor, a proper mechanosensor in the ER? And just to tell you how I define what a mechanosensor is, a mechanosensor is a protein that converts a mechanical stimulus into a biochemical stimulus. Like the best characterized would be the piezo channels, which are channels that will open once the cell is, or the membrane is exposed to mechanical stress or force, and then they would initiate a calcium current, okay? So the question is now, is the ER itself involved in mechanosensing? And this is an area that has been really overlooked. There's so many known mechanosensors at the plasma membrane, like the piezo channels. There is one mechanosensitive protein, which is cytoplasmic phospholipase A2 at the nuclear envelope. But other than that, organelles have been overlooked with respect to mechanosensing. And we thought the ER being a compartment that stretches through the entire cytoplasmic area, forming contacts with the plasma membrane and all the other organelles, it is actually predestined to be a mechanosensitive organelle. So in order to start, looking into this, we took an educated guess, and we thought that maybe RA1, this is a transmembrane ER resident protein that senses normally proteotoxic stress. So RA1 is activated by proteotoxic stress and then initiates splicing of XBP1, which then initiates the unfolded protein response. It's part of one of the three branches of the UPR, the unfolded protein response. So RA1 is a kinase and has also this nuclease activity. So it has two things. So one way to look at RA1 activation will be to look at autophosphorylation of RA1. And normally this phosphorylation is a trans autophosphorylation. So RA1 will form a dimer, and one partner in the dimer will phosphorylate the other one. Okay, so you need a dimerization in order to see RA1 phosphorylation. This will be important in order to understand this. So then why did we look at RA1? Why was our educated guess RA1? The lab of Robert Ernst actually showed very nicely that RA1 can be activated by lipid bilayer stress. In the absence of any protein toxicity, there are domains within RA1 that can sense lipid bilayer stress. So if the ER membrane is not super happy, if there are packing defects, then RA1 will be activated in the absence of any UPR. Okay, so this we thought maybe mechanical stress will induce some form of lipid disorder. This is a paper by Mathieu Piel. This is the model that they had in this beautiful paper from 2020 showing that the nuclear envelope, when the membrane is stretched, exposes some packing defects which lead to recruitment of CPLA2, which is like a nuclear envelope-based mechanosensor that will induce different cellular phenomena. So we thought maybe RA1, if we expose cells to like compression stress, maybe the ER membrane will be also undergoing some form of lipid bilayer stress, and this will activate RA1. So how do we induce confinement of the cells? We grow the cells on a like glass cover slip. Then we come with pillars, and these pillars have a certain height. So for instance, these pillars have a height of 3 micrometer. So this will determine the height that the cells are allowed to grow. So cells are normally, like for instance, HeLa cells, which will be most figures will be done with HeLa cells, have a height of about 10 micrometer. So if you compress them down to 3 micrometer, they will be confined to 3 micrometer, and this will be the mechanical stress. So when we come down, some of the cells, and this is about 20% of the surface area of the cover slip, will be squashed. So the cells under these pillars are dead. You have to live with this. But the majority of the cells on the cover slip will be confined to the height that you have. That is the only way how you can do these confinements with this technology. And if you want to see how the ER behaves, these are cells that stably express Sec61 beta. This is an ER marker. And when you confine them, this is just in seconds, you see really that the ER undergoes also structural changes. Yeah. And we actually know that the ER undergoes a transformation from tubules to sheets. So there's a sheet proliferation in cells that are experiencing confinement and compression as well. So we thought that is RA1 activated. So this is the first figure. So this is the basal level of RA1 phosphorylation. This is the positive control with tapsigargin just to activate the UPR. And you see that within about 10 minutes, you see the first traces of RA1 phosphorylation. And then as time goes on, it saturates at about 30 minutes. And then you can keep the cells for 3 hours in confinement. You will not get more RA1 phosphorylation. So at about 30 minutes, you reach a plateau, and then it stays like that. I'm not showing the data when you remove the confinement. The RA1 will stay phosphorylated for another 30 minutes, and then it will be dephosphorylated and going back to the normal levels again. So RA1 undergoes phosphorylation activation when cells are experiencing confinement. This graph is shown in HeLa cells. We did it with 8 different cell lines: transformed cells, non-transformed cells, primary cells, even non-adherent cells like multiple myeloma cells. When you confine them, they will show this RA1 phosphorylation. So this is a very broad response that we see. So this is just in HepG2 cells, RPE1 and fibroblast. You see always this phosphorylation, different degrees, of course, depending on the basal level of RA1 that is expressed in these cells. So RA1 then responds to mechanical stress. The question is now, is it downstream of any known mechanosensitive pathways? So we started with piezo. We treated the cells with gadolinium to block piezo channels, and we still see RA1 phosphorylation that is not distinguishable from controlled DMSO-treated conditions. So blocking piezo channels does not prevent RA1 activation, indicating it's not downstream of piezo channels. Is it downstream of PLA2, which was known to be an organelle-based mechanosensitive molecule? It is not. When you block CPLA2, you still see RA1 phosphorylation that is almost at the same level as control treated cells. So RA1 is activated independently of CPLA2 and is also independent of TOR, ERK, ABL, and AKT, which are all kinases that are involved, activated downstream of compressive stress. So whatever stimulus we used, we never see that RA1 is dependent in its activation downstream of any other molecule, indicating that it might be really sensing the mechanical stress itself and not acting downstream of other mechanosensors. Then we asked which cytoskeletal element is involved in transducing the force from the plasma membrane to the ER. We looked at actin. It was not actin. With nocodazole, when we depolymerize microtubules, we actually consistently see more IRA1 phosphorylation. We think this is because maybe the cells are somehow softer, more fluffy. This is why they respond more with IRA1 phosphorylation. But look what happens when we kill or interfere with intermediate filaments by vimentin knockdown. You see a strongly reduced IRA1 phosphorylation, indicating that the transmission of the force to the ER is somehow dependent on intact intermediate filaments, but not on microtubules or actin cytoskeleton. So that is the conclusion that we have so far. Before I go to the next part of the talk, that IRA1 is activated in response to mechanical stress and confinement, and that the force transmission is mediated by intermediate filaments. And we think that IRA1 is a mechanosensor because it's not downstream of any other mechanosensitive molecule that we looked at. So then the question is, the obvious question is this UPR? And I will go very quickly through this. It's not UPR. So we looked at the PERK pathway just to see if anything is there. This is the positive control for phospho-EIF2 alpha, which is downstream of PERK. If anything, we see a dephosphorylation. So we see less PERK signaling. Yeah. We looked also whether there is a protein misfolding using a dye that starts fluorescing when sulfhydryl groups are exposed. And so it's a marker for misfolded proteins. Also there, when we confine cells, we don't see a sign for protein misfolding. And we also don't see a sign for XBP splicing. Remember, IRA1 has this nuclease domain that splices XBP if this is a UPR-dependent signaling pathway. This is the positive control with tapsigargin, 3 hours. You see very, very nice splicing of XPP, and you barely see any splicing even 3 hours after confinement. So there is really no UPR, no protein misfolding that we see. So this is a novel, non-canonical way of IRA1 activation. So it's not UPR. So the question is now, is it lipid bilayer stress? So how can we show this? We took two approaches. We took first an approach using FRAP. So we bleached the ER in the periphery and in the juxtanuclear region. And I will just show the dynamics that we recorded from the juxtanuclear region. In the periphery, it's the same. The half-time of two transmembrane proteins, calnexin and Sec61 beta, is faster so the half-time is lower, so which means that the fusion is faster. So the fusion of transmembrane proteins in the ER membrane is faster when cells are exposed to confinement. So the way how we do this experiment, we confine cells for about 15 minutes, and then we start recording and this is what we observe. So this is a sign that something is wrong with the ER membrane. Something is changing with the lipid bilayer, allowing for faster diffusion. And then we made use of this reporter. This is a bacterial protein that is known to be mechanosensitive, that was engineered to contain a circularly permutated GFP. So when it stretches, the GFP stops fluorescing, the fluorescence decreases. And you see here very nicely that when we expose cells to confinement, the fluorescence decreases by about 25 to 30%. So, and that is in the range that they observed in their original paper, the decrease of fluorescence when they exposed the membrane to a stretching stimulus. So we think the ER membrane is undergoing some changes, some mechanical stretching that might be linked to IRA1 activation. So can we test this more directly? So we made use of two mutants of IRA1, one in the transmembrane domain, one in the amphipathic helix, that are known to still respond to tapsigargin. So when you induce UPR, they are still phosphorylated. But look what happens here. While IRA1 is phosphorylated in confined cells, these mutants of IRA1, and this is not overexpression. So we CRISPR mutated the endogenous IRA1 copy in these cells. These are hex cells, by the way, that respond this way. So you really see that there's no activation at all, not less. There's no activation. So these mutants, although they still respond to UPR, they don't respond to mechanical confinement of the cells. So we think that IRA1 activation is by lipid bilayer stress. So the question is now, why does the whole thing happen? Like what purpose does IRA1 serve when it's activated. If it's not UPR, what does it do then? And we made use of this paper. This is a really beautiful paper in a journal that I normally would not read, Circulation. I found it actually by accident. And they showed that cardiac hypertrophy is mediated by RA1 activity. And this is an activity that's independent of the splicing of XPP. So it's a non-canonical role of RA1 that regulates protein synthesis and therefore the hypertrophy of cardiac myocytes. So, and they showed here in this paper that RA1 interacts with EIF3B and 4G to increase protein synthesis. And we thought, okay, does confinement regulate protein synthesis? And this is like the first experiment that we did. So this is a sunset assay. So it's a puromycin incorporation. The more the cells do protein synthesis, the more they incorporate puromycin, which you can stain and then detect by FACS. And you see here a very weak, really barely noticeable decrease of translation in cells that are exposed to confinement. That's the red curve here. When we inhibit RA1 in non-confined cells, we see a weak inhibition. So you see this is more noticeable that there's a population of cells that are not translating very well. But what happens if we combine RA1 inhibition using this chemical inhibitor with confinement? The cells essentially stop translating. So translation under confinement is addicted to RA1. Of course, this is a drug that could have off targets. This is just a quantification of 4 to 5 experiments. We also did it in RA1 knockout cells, and RA1 knockout cells also stop translation when they are exposed to confinement. These are knockout cells under normal conditions, and this is the confined cells when they are pretty much like the chemically inhibited RA1 cells. So again, we conclude that translation under confinement is addicted to RA1, and RA1 prevents the decrease of translation under confined conditions. So this is one model that we have. So we think that confinement activates RA1 through bilayer stress. RA1 activates translation. I will keep this question bar. I will remove it in a while. The question is, why does confinement inhibit translation? And our guess was based on these two papers. One paper showed that confinement reduces the cell volume, which we confirm in our own data, that there's about a 30% reduction of cell volume when cells are confined. And this paper by James Farrell's group showing that the viscosity of the cytoplasm determines how translation works. Yeah. So if the cytoplasm is more crowded, the cells will not translate as well. And we thought, okay, can we show that the cytoplasm is more crowded? So we used this GEM trackings with help from the lab of Liam Holt. Beautiful collaboration. They helped us a lot getting this method working. So these are small spherical molecules that you can express in the cells that attack with GFP. They have a diameter of 40 nanometers, which is about like 2 ribosomes. And you track them for about like 2.5 seconds in the cytoplasm, and then you can measure the diffusion coefficient. I think it's very clear here that confined cells, you have this left shift, which means there's more cells that have very slow diffusion and fewer cells that have faster diffusion. This is the calculated diffusion coefficient, which is also in the range of what you theoretically would assume if you would see a decrease of the cell volume by about 30%, you would expect a 25% to 30% decrease of the diffusion coefficient, which is exactly what we see here. So we think that this is the confinement inhibits translation through crowding. And the question is now, how does RA1 regulate translation? Of course, I told you that it binds EIF3B, but what does it do under translation? And this is RA1 stably expressed in cells when you confine them, which you see by blebbing of the cells. This is just a sign that they are confined, that RA1 forms clusters. And the funny thing is these clusters are very rigid. When you bleach them in a FRAP experiment, you see here they don't recover even after 5 minutes of post-bleaching. So they never recover. And by the way, these clusters they can be dissolved. Once you remove confinement, within about 20 minutes, the clusters are gone again. Although they don't recover in a FRAB experiment, they can be actively disassembled once the mechanical stress is gone. And the question is now, do these clusters co-localize with these initiation factors for translation? By the way, these clusters are very different from Tapsi-Gargin clusters that Peter Walter showed. We can reproduce these. These are very large. They are very different from these very small clusters that we see here. And we see here a co-localization. This is just a few examples of RA1 and EIF3B. The 4G antibody didn't work very well, so we couldn't make it. But with this factor that was known to co-immunoprecipitate with RA1, there is co-localization on these small domains of the ER. So I will skip this slide, keep it for the discussion. So this is our model. We think that confinement activates RA1, and RA1 prevents the decrease of translation that is imposed by confinement. So with this, with the summary, we think that RA1 is an ER-based mechanosensor that senses lipid bilayer stress that is induced by mechanical forces, and it's independent of the UPR. It's a novel, non-canonical role of RA1, and the physiological function is to maintain translation. The whole work that has been done entirely by a tandem team in my lab. So this is Louis, a postdoc in my lab, and this is Michi, who is the major driver of the project, a PhD student in the lab. So they work together on this project and have made incredible progress within just 2 years. This is the data that we accumulated. Thank you very much for your attention. I'm happy to take questions.